welcome to my channel. This is the 8th episode of the Meet the Pro series, a series where I will be interviewing professionals in the field of biology and genetics to help high school students interested in biology. In today's episode, I will be interviewing Dr. Constance Cepko. She is the Bullard Professor of Genetics and Neuroscience at Harvard Medical School, studying developmental biology in the retina, and I am delighted to have her with me here today. Thank you for being here, Dr. Cepko.
Sure, my pleasure.
So, could you take a few minutes to kind of describe how you became interested in biology to eventually getting to where you are?
Sure. So I was younger than you, actually. When I was in middle school, so I grew up in Maryland, and there we had to do science fair projects in middle school, and those were presented at a science fair. So I had to do one. Wasn't even that interested. I didn't know what to do, and I asked my science teacher, and at that time there was a magazine, sort of Science for Kids, and she gave it to me, and I looked through, and I saw one of the projects was growing yeast, which I thought was really interesting and accessible. So my little science fair project had to do with growing baker's yeast. It's the same yeast that you would make bread with. So I grew that in my mom's kitchen, and had different media that I grew it on, kind of interested in whether it grew better in sort of honey or sugar or other things. And I got pretty intrigued. And my judge at the science fair, John Palmer, asked me if I would like to visit his lab. Since I grew up in Maryland, there were a lot of government labs. So I said yes, and I went and visited his lab, and it was this idyllic setting of a little sort of white house in the woods. It was called the Forest Disease Lab, and the job there was to identify tree fungi. So I met a bunch of people at the lab, and I was loving it. And he asked me if I wanted to come and work there on weekends, so I did. And remarkably, he would show up and teach me how to make media and sterilize things and grow various microorganisms. So I did that for years. So I knew I loved to do research, and then I just followed that path. It was pretty straightforward. I went to University of Maryland, majored in microbiology, then went to MIT to graduate school for a PhD in biology, where I worked in virology, growing adenovirus in Phil Sharp's lab.
Okay.
And then I went on to do more virology, but from the standpoint of viral vector production with Richard Mulligan at the Whitehead at MIT. And then I came to Harvard, and I started my lab in 1985, and that's where I've been ever since.
Did you always know that you wanted to work at a medical school rather than?
No, I didn't. I really didn't understand the different niches where a person could run a lab. When I left my undergrad, so I had been at a big state university, I kind of knew what those were like, and our medical school wasn't integrated into our undergrad. So I didn't know anything about medical schools. And at MIT, there really was very little connection at that time with any of the medical schools in the area. So I really didn't know what it was like. So actually coming here to Harvard was the first time I truly interacted with people who were doing research at a medical school. And I found it to be kind of similar. I mean, people around me, some people do very basic things like yeast genetics, or basic processes of transcription and things like that. So some things that we do in our department have nothing to do with medical applications, and some do. So I found it pretty similar to the other places I had been in terms of topics and process.
Could you talk a little bit about your work on the retina?
Sure. So I had always been interested in developmental biology. And even though, as I mentioned, I was trained in virology, when I was leaving my postdoc, I was trying to figure out what I wanted to do in my own lab. And I thought, I wanted to do something that was unique and open-ended and different than what I had been doing. What I had been doing in terms of making viral vectors was being done in many labs. So I didn't feel the need to continue doing that, and I didn't want to be redundant. So I looked around at things that I thought had really big unanswered questions that were exciting, and I thought about neuroscience. So I knew nothing about it. So I started to educate myself. And because I was interested in development, I started thinking about, well, where did all these neurons come from? And I devised a method to trace lineages in the brain using viruses I had made as a postdoc. And so I started doing that, and I chose to work on the retina initially because it was accessible for viral infections. We subsequently also worked on the brain, and we did that in rats and mice and chicks. And then I discovered that the retina was a much more accessible system for various types of perturbations and studies. So we focused then our mechanistic work on the retina. So we just started to study then how do all these different, in the retina, there's 120 cell types. So it's not a simple camera.
Yeah.
It's more of a parallel processing computer. And I was interested in how you generate all the cellular complexity that allows you to do all the methods, or sorry, all the circuits and ways of extracting information from the visual scene, all of which happens locally in the eye.
Hmm.
So we started studying development, and I'm still fascinated by that.
How did you like track the lineage of the neurons using those viruses?
So what we used were retroviral vectors, and these are viruses that when they infect a cell, their genome is RNA, but they convert their RNA into DNA using reverse transcriptase.
Yeah.
And then the viral DNA integrates into the host chromosome fairly randomly. And once it integrates, it can transcribe, and you can rig it so that it's crippled. It can't spread cell to cell,
Hmm.
but that viral DNA that's integrated into the initially infected cell will replicate along with the host genome and get passed to all the progeny. So it's an indelible genetic mark. And by diluting the virus appropriately, you can have very sparse events. So you can start to ask, if we infect a progenitor or mitotic cell here or there, what kind of daughter cells does it make? And that's the question of lineage. And we could find those cells because we could engineer the virus to express something which allowed us to see the cells that were infected, a dye, a substrate for an enzyme that would turn color, those sorts of things, GFP now. But
Hmm.
at the time, we were using beta-galactosidase or alkaline phosphatase to allow us to see the infected cells. So the retina, again, offered a huge advantage there because the sibling cells, or the cells that were clonally related,
Yeah.
would arrange themselves in a very convenient to analyze way, in a sort of a radial array across the tissue. So there was no doubt about who was related to whom. When we did that experiment in the brain, the cells would migrate all over the place, and we had to invent barcodes to track them. And actually, we made the first barcoded libraries in 1991. So 20 some years before other people made them.
Hmm.
And it worked great. We could track individual single cells anywhere they went in the brain by analyzing the viral encoded barcode. So that's how we did it, and we still do the same technique, looking at lineages.
I was reading like on the lab website the other day, and you mentioned that the retina is, to quote, a tractable model for the rest of the central nervous system.
Yeah.
What does that exactly mean?
So the retina is part of what's called the central nervous system. So these are the areas of our nervous system that develop from the neural plate and then the neural tube. And if you look anywhere in the nervous system, really what you see are cells that receive information.
Yeah.
And then there are local cells that process the information. And then there are cells that give output to other areas of the body or the nervous system. And so the retina has all those elements. So first of all, it develops from the neural tube. It has cells that receive information. In the case of the retina, it's light. It's a sensory system. So cells that receive information are rod and cone photoreceptors. And then those cells communicate that information to local cells, interneurons, which basically discuss the input. And then there's output cells called ganglion cells, and they send their axon from the eye, so directly from the eye to many target locations in the brain. And the interpretation of what we see is done in the brain. But the first steps of processing, for example, the direction of motion or edges in the visual scene, those things are all processed locally in the retina. And then perception happens in the brain. And so these are basically the same things that happen everywhere in the brain.
Yeah,
Again,
yeah.
information is received, it's processed, outputs given. And, again, the type of information received, where it comes from, differs depending on where you're looking in the brain. The sensory systems are pretty straightforward because they're sensory cells where the information is first received.
Hmm.
But in the brain, many places in the brain, inputs arriving from a lot of different places, including the sensory systems. But it's more complex in terms of the input-output relationships.
Information kind of seems like I guess information flow seems to be like a big theme here in that part. And this is like something I've always wanted to know the answer to, but are there sort of like basic principles with how like we learn?
Oh yeah, and people are trying to establish those.
Yeah.
The idea mostly is, and some of this is from model organisms, is that we can change the strength of synapses. So synapses are the area between two neurons that are talking to each other through neurotransmitters.
Hmm.
Those structures can strengthen or weaken depending on the type of input or the um, amount of input or the
Yeah.
frequency of input that might use those synapses. So one idea is that things that are repeated, let's even just say a motor program, things that you're
Hmm.
learning through motor activity, those will develop sort of repetitive transmission patterns among neurons and neurons into muscle. If you do that enough, you can strengthen them. And this is actually protein interactions or protein depositions or protein structures that are built at those synapses that can be stronger or weaker, depending again on use. And so that's one fundamental aspect of how we learn. I think a lot of how we learn and how we store memories,
Hmm.
still big questions. I mean, and I'm not an expert on that area, so you probably could find a better neuroscientist to talk to about that.
Okay. All right, so now I guess I'll go back to kind of like more around your research. Uh, since like some of your work revolves around fighting degenerative diseases, like especially with blindness, uh, have you ever like was stem cells like ever like a big part of that? Especially like kind of to counter the cone or RPE cell death?
Yeah, so when stem cells were becoming really popular, I guess, we've known that there are stem cells for a very long time, but stem cells in various tissues, including the retina, were then sought. And in the retina itself, there have been reports of some stem cells in mammals. Mostly we know there's stem cells in fish
Hmm.
and other organisms, but they really haven't been discovered in the adult eye in humans or other mammals. But you probably know that you can make stem cells from fibroblasts or skin
Yeah.
tissue. And then from those stem cells, those are called induced pluripotent stem cells, you can grow a retina. And
Hmm.
people are growing retinas from those iPSCs,
Yeah.
and they would like to use them therapeutically. There have some of the first transplants of stem cell derived cell types have been for the eye. So the eye, I think you probably know, has an epithelial cell type called the retinal pigment epithelial cell type,
Hmm.
or RPE. And in certain diseases, the RPE suffers and dies. And RPE cells grown in the lab from stem cells have been grafted into people now.
Okay.
The jury's still out. How well do they survive? Do they function? Does a person see better? I don't think we know the answers to any of that,
Hmm.
but it seems safe. So far,
Yeah.
so good on that.
Yeah.
Um, People would also like to transplant in photoreceptors. So photoreceptors, rods and cones, are the ones that die in most of the diseases. And those are harder because when they get grafted in, they have to form these synaptic connections with special neurons called the bipolar and horizontal cells. It's not clear that will happen. There's some data from animal models that suggest that that can happen. I'm not convinced. And I'm a little worried about two things.
Okay.
One is that a diseased eye presents a very challenging environment. So in any eye that's degenerated to the point where you would want to risk a graft, and there is risk involved with these things. For
Yeah.
example, will the cells keep dividing and make a tumor is a big risk.
Hmm.
Um, so by the time you actually want to inject cells into someone's eye, it's a degenerated eye.
Yeah.
And the degenerated eye has many challenging aspects to the environment. There is usually oxidative damage. There usually is inflammation. There's something called a glial scar, which
Hmm.
is the growth of processes from glial cells, which tend to block the normal processes that go on, including synaptic formation. Um, so I'm not sure that putting in cells and asking them to synaptically connect with the correct partners is going to work, or
Okay.
that they'll even survive. So again,
Yeah.
the environment is noxious. Now the RPE cells are much simpler. They don't need to form synapses. They just form an epithelium or tight junctions with each other in
Yeah.
a monolayer. That's a much lower task or lower ask of cells than finding the right synaptic partner, forming a meaningful connection in a very scarred bit of tissue. So I'm not sure that the photoreceptor grafts are going to work, but people are working away on it, and maybe, I hope so, but
Okay.
I do worry about those aspects.
Yeah. Now, is there like any sort of like a scientific achievement that you've had in like your career that you would say you're most proud of?
Um, yeah, I think developing the lineage tracing tools is
Yeah.
something that I'm proud of. The first lineage tracing done with viruses in clonal fashion in vivo was from our lab, and then the first barcoded libraries to achieve the same thing was from our lab. Those things I'm pretty proud of.
Hmm.
Um, there have been a lot of other things along the way that I'm also proud of, but those are probably the easiest for me
Yeah.
to suggest.
Okay. And finally, I want to end with like a sort of fun question. If you could bring back any extinct species, which would it be and why? And you cannot say dinosaurs.
Okay. I don't know if I'd want to bring them back either. If you
Okay.
saw the Jurassic Park, you'd have to wonder about that.
Yeah, yeah.
I've always thought it was kind of crazy that people talk about passenger pigeons. And I know this might sound very weird, but apparently there were so many of them, like in New England, they would actually darken the sky when they would fly overhead. And I always thought,
Okay.
boy, it's really sad that we would kill the entire species if they were that common. And they must
Okay.
have been a chunk of the food chain too, if
Hmm.
they were that common.
Yeah.
I don't really know what part of the food chain, but it always sounded to me like, how could we do that? How could we take a species that was that abundant and totally decimate it and make it extinct?
Okay.
That's just off the top of my head. I have to
Yeah,
think about it a little more
that's
to
fair.
really know a better answer. But that
Yeah.
one always struck me as kind of astonishing that we would
Yeah.
do that.
Okay. Well, I mean, I think that's all we have time for today. Thank you, Dr. Cepko, for the interview. I really appreciate it.
Sure. And take care, Ruchi.
All right. Thank you.
Nice to have you in the lab.
Thank you. Bye-bye.
Yep. Bye-bye.